Hello, everyone. My name is Robert Grass. I'm a member of DDAX, which is in DigNA, one of the consortia within the portfolio, working on applications of DNA data storage. It's my great pleasure today to introduce you to our two panelists, to our two speakers of today's webinar on sequencing. They'll hopefully give us a really in-depth view into the sequencing world. First, we'll hear from Laurent Farinelli.
Hello.
He did his PhD at the University of Geneva and then worked in Geneva at Glaxo Wellcome Institute and co-developed part of the Illumina technology, especially the amplification that happens at the beginning of the in the Illumina machine. He then went on to co-found the startup Mantara Predictive Medicine, where he developed further parts of the Illumina technology, for example, the Y-shape adapter that many of you might know that we use in ligating adapters to our amplicons. This company was then over several steps sold into Illumina. Laurent then founded his own company, Fasteris, which was the first company in the world to offer Illumina sequencing services. So really, many of us use sequencing service, was one of the first companies. This company was then sold into Genesupport, and now they're offering services to many customers, mainly within Switzerland. Our second speaker, he's more from the, let's say, current sequencing technology world. He did his PhD from the Technion in Israel, I think from Zohar's group, if I'm not mistaken. He then was on the team of Foundation Medicine, which developed a genomics profiling assay. And this technology is used today to generate millions of patient reports. And he then joined Ultima Genomics as a chief scientific officer with the goal of bringing the world to $100 genome sequencing platform. And so he'll follow after Laurent. With this short introduction, I leave it to Laurent to bring us into, let's say, give us a glimpse into the history of the Illumina sequencing world. Please go ahead, Laurent.
Thank you, Robert. Thank you very much for the kind invitation. It's been a long time since I gave a scientific publication, so I had to dig for historical slide to give you this perspective from a long time ago. So we'll start immediately. It was coming back from a postdoc in Brazil to Geneva in Switzerland, where the head of the Glaxo Wellcome Research Institute told us not only to have fun in what we were doing, which is very important, but also for pharmacogenetics and pharmacogenomics, hoping to find the right drug for the right patient. We were working on a project that was taking years and over 60 people to sequence a small 1 million base genome, Streptococcus pneumoniae, with 7 ABI instruments. It was a tremendous work. And then I was asked with a colleague, Pascal Meyer, to develop the faster sequencing technology to indeed find the right drug for the right patient. And we learned about Sidney Brenner's idea of massively parallel sequencing, and we didn't do what we were expected to do. We found a way to do something differently based on this idea. And trying to also avoid patents, we decided to find 2 modules. One for amplification of DNA, replacing the bead, which was very brilliant but highly complex as well. And the other, it was for base-by-base sequencing, replacing this restriction enzyme-based sequencing developed for MPSS. It was a very special moment, and at Glaxo we had notebook. We were supposed to write it, and this I made sure I wrote it in my notebook because we came with ideas. On this special day, I would start a sentence, Pascal would continue, and we knew it was very emotional, very strong moment, and we knew we invented a new engine for molecular biology. So it was the so-called PCR colonies with bridge PCR on one side, one of the module, and the other was this base-by-base sequencing. So in 96, that's already a long time from now. By Christmas, we're very lucky, and certainly as young scientists, to have a wonderful present, we looked at the microscope, and it was like looking at the stars in a beautiful summer night, except the stars were green because we're using cyber green, and that's where we had the proof of principle that we could generate PCR colonies. About the same time, I performed sequencing technology with only adding one base at the time, with the cycle which is of the nucleotide which is added, and we could generate up to about 40 bases, proving that we could do step-by-step sequencing. And so we filed 2 patents, one for each of the application. And you can see here on the patent the bridge technology to generate the DNA colonies. And on the other side, the step-by-step sequencing with increase of signal depending on the base which were added. We also designed a kind of device that could be used for this. So unfortunately, Glaxo closed the institute in 1997, probably for fiscal reasons, and it was taken over by Serono. And in 2001, with our colleagues, we decided to start a company which was founded by Serono. We named it GenInEx, Genetic Information Extraction, and it was renamed later Montea Predictive Medicine. We developed incredible things as well in this company. The basic idea was to have your own genome on a CD. That was the storage at the time. And having like a clinical trial with, imagine 6,000 patients, 2,000 for each of 3 disease conditions and response to treatment. And when you have your genome on a CD, you go to the doctor, and it's like a fingerprint. It would be able to say you are in group A, B, or C for the response to treatment. And without knowing the sequence itself, you could nevertheless predict what would be the best way to treat the patient. And at the time, I had the idea to introduce a Y-shape adapter, which enables to have 100% when you do a ligation of, let's say, A and B adapters on the same DNA molecule. Instead of having a single adapter, a normal adapter, it would be 25% AA and 25% BB that would be lost. And this adapter here in red and blue, my colleague Magne Osteras, he complained a couple of years ago, why did Illumina give this stupid name of P5 and P7? And then he found his own notebook, and it was himself who had given these names. It was sequences that we developed trying to avoid concatenators, because if the adapter would ligate to each other, it could automatically generate some spots. And we tried to find some ways to avoid this. And that's how we came with the P5 and P7 adapters. We as well developed the instrument itself, a microscope with a flow cell, which was later used by Solexa. We also developed reverse terminator sequencing chemistry. Our colleagues were trying to add Alexa fluorophore to a base with a cleavable linker, and they complained that they could not extend by more than 1 base because the polymerase was blocked. And Magne reacted, say, wow, that's great, because when you cleave the linker, then you recover a normal sequencing process. And that's, even if we don't know, is probably what was used then later by Illumina. We also developed at the time a software for real-time DNA colonies detection and base calling. The company, because also it was a startup, and if you have a startup, we had the CEO, and CEO wanted to be, let's say, the Microsoft of biotechnology, and all the mistakes that could be made were made. And the company was sold by Serono without really understanding the potential for 2 million in shares and 2 million in cash to our competitors, Solexa in the UK and Lynx in the US, who later merged under the name Solexa and were acquired by Illumina, using our device, for example, to demonstrate sequencing on small genomes by $600 million. And that's about the time where we acquired a Solexa instrument at Fasteris. Now, the sequencing technology, the goal was exactly like you have bacteria, you open a petri dish and you incubate it overnight. In the evening before, you don't see anything with your eye. In the morning, you see spots because the bacteria have amplified. And that's exactly what we obtained with the in situ PCR. You have here different DNA sequence coming from different parts of the genomes. And then with the in situ amplification, you can see with 10x object microscope, you can see the spots like we did before. So you have about 1,000 time amplification of a single molecule into a cluster of molecules which are located physically next to each other. And how to do this, which is a tremendous density. On a fingernail, you can have up to 10 million DNA colonies. You clone your DNA. So first of all, you break it. Could be physically, could be enzymatically, and you put the P5, P7 adapters at the end. And then you have the functionalized surface. We were using nuclear link on which we covalently attached the P5, P7 adapters. And you hybridize with your template molecule, which were denatured, and you have it on the surface. The adapters are linked by the 5' end, covalently attached, and of course the 3' end is free, ready for primary extension. And that's what we do as a first step. We use a polymerase, we extend it, and we denature, and therefore you have physically attached on the surface your template. And that's the most important part, to have the right density. If you have a too high density, the spots will be too close to each other and you will have a limited number of sequence. If it's too far away, you will have not enough spots and not enough sequence at the end. And that's when you do service, it's very important to be sure that you have the right concentration of the library you put on the surface. So in practice, you have your, after this first step, you have your template. I represented here only one. And you have this red adapter, which is ending with a 3' end, and you anneal it, and it will look for the corresponding adapter on the surface. And because on the surface, the corresponding adapter is covalently attached by his 5' end, the 3' is ready for extension, and that's what you do, doing a bridge PCR, let's say. And when you denature, physically, the copy is next to the original molecule and not like in a liquid solution when everything is being mixed. And you do the cycle a couple of times. And at the end, you have with a PCR about 1,000 molecules per spot. Now, for the base-by-base sequencing, you first start with a polymerase extension with a 4 nucleotide. Each is labeled with a different color and blocked on the 3' end, maybe because of this cleavable linker. And therefore, only one base is added on each DNA colony. And if you do image acquisition, you take a picture and you can see in the 2 spots that are represented here, the first base is green. So it means that it's a T. Then you deblock, like if you remove the cleavable linker, and you're ready for the next step as if it was nothing was done before. And on cycle 2, you see here that the green has become blue, so you can have the base calling from T to G when the other is green and is T and T. Chemistry is quick, about 10-15 minutes, and what takes time is taking the picture, at least at the time of the first instruments. And then you continue the step and each of the base, and you can have the base calling based on the color on the DNA clusters here, and you can read the sequence. On the bottom, you can see that when you have homopolymers, it works very well because you can really count the bases in contrast to other sequencing technology. You also have to do the bioinformatics. So you generate a text file with X, Y position and intensity. And you also have to filter out, for example, location where you have two spots which are too close to each other, and it will give a mixed sequence. Now to the sequencing speed. When we invented the DNA colonies in 1996, I calculated it was about 4 base per second with 7 ABI sequencer instruments. Then a capillary instrument, not only a glass plate. In 2003, when we founded Fasteris, it was about 10 base per second. And when we acquired the first Solexa 1G instrument in 2006, the speed was about 300 base of second, which is an incredible increase compared to the previous technology. It's really rare that you have such an increase, which is over 10 times more. But it was just the beginning, because already in 2007, there was a paired-end sequencing, a new pipeline of the genome analyzer version 2, and you can see you get up to 50,000 base per second, close to 2009, 2010. And it was only the beginning, because, of course, in 2010 came the HiSeq, and all what seemed to be very high in terms of speed is really at the bottom of the scale. And you have, with the HiSeq sequencing technology, we go close to 1 million base a second in 2010, 2011. But of course, as you know, then came a new HiSeq instrument in 2014, going to 2 million base per second. And then I stopped in 2017 with the NovaSeq, which was close to 20 million base per second as a sequencing speed. Well, in 2003, as I told you, we founded Fasteris. As we were in Switzerland, instead of selecting a garage to start our company, we decided to found it in a chalet. And you can see my colleague Magnus Torras and myself acquiring a second-hand Sanger sequencing instrument. And together with Anschendorf, that's where we started our operations. We had the installation run in our company. It was, at the time, with a Solexa instrument, 8 lanes of 500,000 reads of 20 base each. And it was very difficult for me to analyze the data. I had to use a MySQL database to sort the reads. And it was almost easier for me at the time to use DigNA, which was developed by the Schrenzel group for de novo assembly. And that's one of the first view on the mapping of the reads. That was the way to prepare the libraries. We had these kits from Solexa, the Solexa cluster station that by mistake I thought it was a computer that was coming together with the system, and then I learned that it was not a computer cluster, but generating the DNA cluster that we were naming DNA colony at the time, and the instrument itself to generate the reads. We developed different applications very quickly in 2007 to analyze whole genome, de novo assembly of transcriptome, ChIP-6, and also small RNA. At the end of the year, we're very, very proud to be considered by Illumina not only the first service lab company to acquire the machine and the smallest lab to use the technology, but also the lab who developed the broadest range of applications. Application that we were doing was for whole genome, for ChIP-6, small RNA, transcriptome, and also medical analysis. And I will jump quickly on some of these applications, but before that, I will go for the sample preparation, which is more technical and for this seminar. So first of all, you break your DNA. You have to do end repair with DNA polymerase, Kleno, and then you have blunt end. You add an A with a Kleno exo minus, and you have an overhanging A to prevent self-ligation of the fragments. And then you ligate the Y-shape adapter, which have a T at the end. And when you have this ligation done, then you have all your fragments who have A and B, or P5, P7 at each end. You then if you take the example of a single molecule, I represented here the two sides of the insert. One is in yellow and the other in green. And you can see that next to the, let's say, P5 side, you have on one molecule the yellow and the other is the green. So with a single template, when you do the first PCR reaction, what happens? You have these two strands which are denatured. And when you do the amplification, then you have these two types of molecule, one with, let's say, the yellow next to the blue adapter, and the other with the green next to the blue adapter. And you have, of course, then when you sequence two different reads which are obtained, you do the DNA clustering with the linearization, blocking, primer hybridization, because, for example, when you generate the DNA colonies, you have to remove one of the two strands here, those with a red adapter covalently linked on the surface, and what is remaining giving you two different types of reads from the molecule, from the two ends. Some example of application, we've developed a ChIP-seq protocol, and we had to pray for this test. It was mice losing memory with age, and when you would put acetylation of histone in a normal way, they would recover memory. It was embargo for the news, and in the morning when I expected the publication to be published, they just spoke about it on the radio. I was excited, but then it was something else, which was also from the same technology. It was a Neanderthal genome that was done by other researchers on Illumina sequencing technology. Developed with a doctor in Italy a way to do whole exome. So before that, when they were looking for a mutation, it's like on a room which is dark, and you use a spotlight to look at one gene or another. Is it this one? Yes, no. This one? Yes, no. Here, we took the whole exome. It's as if we switch on the light and we look at everything, and then we look at the difference between the cancer and the healthy, and we could find that it was a BRAF mutation that was not supposed to be in liquid cancer. We sequenced like an genome. We had to use haploid male individuals together with the University of Lausanne and Swiss Institute of Bioinformatics. Very complicated to assemble the genome with a lot of copies which were identical and insertion in the genome. Very small spider and transcriptome, analyzing on those animals which have a broad range is a pest from broad range of plants. And interesting application, it was customers in Peru who had the idea to do a de novo assembly from the small RNAs, and they could detect not only the virus that they knew had infected the plant, but also three genomes of novel virus that they didn't know. And the technology was used by a quarantine institute in Australia and USA, sending us sample to analyze possible new virus coming into plants coming to the countries. Also, clinical metagenomics that we developed to find a clinical grade method from the first publication we had made together with Schrenzel on Illumina for microbiome genome. Finally, a slide from a couple of years ago in the company then Fasteris, and in 2020 we renamed the company Genesupport after the integration into Medisupport, which was then renamed Sonic Switzerland, and going also in the medical field in 2015. Medical application, NIPT, a test that we developed together with the Swiss Institute of Bioinformatics, somatic cancer panel for tumor, detecting the treatment, germline, the prevention of cancer, also pre-implantation genetic testing, very sensitive because you need special authorization for that, and also very proud to be with accreditation. Genesupport now has 5 locations in Switzerland. We are integrating. My job is to integrate these different companies, merging them into a single one. And we work in different areas, doing medical analysis, medical genetic tests in different parts of human life. We have also medical doctors who do genetic counseling, very important for our patient to give the proper advice. And 2 applications that we are we have launched 6 years ago, together with the partners, is for predicting the risk of developing a breast cancer and adapting possible monitoring before, or prostate cancer for men. And I will conclude with a few slides, they were written in 2020. We're looking at the future, but we see that with the speed and cost, it's becoming cheaper to generate the data than to store it. And that's maybe the topic of the seminar. So I thought it was appropriate to speak about that. Incredible growth in medicine, which I suggest would change the logo to something more like this, DNA instead of the symbol of Hermes. And then also very important, the challenge of accreditation, which is very important for diagnostic. And a concluding remark, which was before the hype of artificial intelligence, but at the time I was already mentioning that we have a changing role for doctors. Now is partnership with patient important of the human experience, but also screening everything, what is the right not to know. And I will conclude here that it's very important for us to apply this technology for the good of humankind. And I will stop here.
Very fascinating. Thank you. The questions at the end, Robert, right?
Yes, yeah. Question at the end. Let's continue in, let's say, this aspect of a timeline in sequencing technologies. Doron, please go ahead.
Thank you, and again, thank you for inviting me to speak at this webinar. So I'll kind of carry the baton forward and talk about kind of what's happening in the future. The future is now. As Robin mentioned, I was at Foundation Medicine working on oncology applications, Ultima, on next-gen sequencers, and now kind of advising a few different companies in the next-gen diagnostic sector. Okay, so the reason a lot of us are interested in sequencing is because we believe that most human diseases have detectable genomic markers. It's obviously true for genetic disorders and oncology, also for infectious disease, but we also believe it's actually true for almost any other disease like autoimmune, etc., and across many different modalities of the disease, from early detection, being able to find and detect diseases earlier, diagnosis, partitioning diseases into different modalities and subsets that can be treated differently, monitoring treatment, and so on. And sequencing is not just limited to sequencing DNA. Obviously, there's genomics, but there's also transcriptomics, proteomics, which is many times measured through sequencing, epigenetics, microbiome, etc. So many different modes of sequencing, and we can all use kind of the same sequencer to measure all this information. So reflecting a little back, where this started, and there isn't really a talk about sequencing that doesn't include some version of this graph. And when the first human genome was sequenced in the Human Genome Project, it cost about $3 billion. It was all done on capillary sequencing from ABI, huge, very long. A lot of work went into this, kind of breaking the genome into small bits and sequencing each one independently. That was kind of the impetus for NGS technologies to start emerging. The first one was 454, which was a bit of a kind of in-between technology, but then the kind of revolution of NGS with Illumina, as Laurent kind of told us about. There are also other contenders, ABI Solid, Ion Torrent platform, and really kind of, the credit goes to Illumina for really pushing down the curve of cost down from the multi-million-dollar point to kind of thousands of dollars over a relatively short time. During that time, there was also another kind of emerging set of technologies, which we kind of tend to call third-generation sequencing, which include very long DNA sequencing, sequence at the single molecule level with PacBio and Oxford Nanopore, both very different technology platforms, which again gave us kind of a peek into another dimension of sequencing. And then kind of, the field kind of plateaued around 2015 or so at $1,000 per genome or so about for quite a while. A lot of work was done during this time to develop different applications in genetics and NIPT and oncology and so on. But sequencing cost was more of a constraint than a driver for kind of for those assays over time. And then more recently, we've had kind of a significant kind of change in this course when Ultima announced its kind of $100 genome a couple of years ago, and Illumina kind of followed with the NovaSeq X at $200 per genome. And then we had Element, which announced a $200 genome, and Roche, which just came out with a new sequencer, and all kind of around the same price point. So we can now say probably around $100 per genome as a baseline sequencing cost for, if you're sequencing at large scale. Important to note that this is the sequencing part. There's obviously all the other parts, the sample prep, the computations, and actually as we generate data faster and at higher throughputs, the analysis part, at least, it becomes a much more significant part of the procedure and the cost. One thing that's really interesting about this specific time point for people doing sequencing is that we've kind of really been evolving from a field with one dominant platform, which is Illumina, and it's had its many generations that cover many different aspects of kind of size and cost and throughput. But now we have actually more specialized platforms. And I chose these four, not because they're the only ones, there's actually a few more, but they actually demonstrate a few different angles of sequencing technologies. And I think the interesting point to kind of think about as we're developing new applications is really that each one has its own strengths and weaknesses. So the prediction is that we're going to move from like Illumina really dominating 80 to 90% of the whole sequencing market to having different platforms, each with its kind of own areas of strength. The Illumina NovaSeq X is still kind of the power horse of the field with high accuracy and high throughput, and most of the large-scale applications like whole genome, whole exome, whole transcriptome are still being done on these platforms. Ultima came out a few years ago, now even dropped its cost to $70 per genome. The main aspect, the new aspect of this technology is that the flow cell, which is kind of the expensive part of the workflow, has been replaced with a very cheap component, and it's also very fast. So it can produce a lot of reads at very high throughput and very low cost. And that's very, very useful for counting applications like single cell RNA. It also has a specific kind of advantage for SNV accuracy, which can be used for applications like MRD. And again, I'm not going to go into all the details of each one of these technologies, which would take another hour. But I want to give you a bit of a glimpse of the different kind of advantages of each one of these platforms. PacBio, which is not a new system, but they've been really innovating and pushing new systems over time. The main advantage is really very long reads. We could actually go to hundreds of kilobases, but using that kind of approach, you can actually sequence the same fragment over and over and get the error rate to kind of very high fidelity reads. So the advantage is that you can create very long reads with very high fidelity. The throughput isn't as high as the other platforms. That gives us, in at least like clinical whole genome sequencing, the ability to sequence all the difficult part of the genomes and really kind of get repeats correct and duplications and so on, which are very important for clinical genome sequencing, and also RNA isoforms and some niche applications like that. And I also wanted to mention the Roche Excellium system, which was just announced last year. It's actually not commercial yet, but it's gaining a lot of interest. It's also very unique in its profile. It's based on a nanopore with a kind of a unique sample prep using kind of a DNA expansion. And this ends up with reads that are about a kilobase or hundreds of bases to a kilobase, which is kind of a bit of a new size range. Has a very, very fast turnaround time. So you can actually create a genome in 4 hours or less on this system. And applications, we can all kind of imagine what you could do with this. Roche is still kind of working with different partnerships on demonstrating these. But I think the interesting part is Roche is a very strong clinical and pharmacological player, so they will definitely be using this platform to push these areas of sequencing. So just to kind of summarize this, we're kind of at a point where different sequencing technologies are giving us kind of new capabilities and different applications that can kind of use each one of these in different ways. A kind of a side effect of this, funny to call it side effect, but it is kind of an output, is that that many, because the throughputs are getting so large and kind of we're looking at many modes of sequencing, and there's many new sample preps. So Laurent kind of mentioned kind of the basic library we all kind of know and love, and people are also kind of using that to develop new applications and sample prep methods for single cell, for example. For example, there's like a few different companies that are developing methods to create libraries at a single cell level. This is at an RNA level, and then there's companies like BioScar, which are also doing it at a whole genome level. And a few years ago, we would have imagined doing whole genome sequencing at a single cell level. Today, with sequencing costs being as low as they are, we can actually afford to do that for a few thousand cells. And we're kind of seeing the interesting kind of diversity of the genome sequence even in a single tissue sample. And people are looking at methylation, obviously many different preps for that, and also ones that are combining genome and methylome, so you can actually see mutations and changes in methylation concurrently. And I also mentioned kind of proteomic sequencing, which is basically taking proteins, tagging them with DNA one way or another, and then using the sequencer to count these tags. So essentially, you're counting proteins through a DNA sequencing platform. And these are just a few examples, but the whole field is kind of coming up with a lot of new ways to prepare samples that are both very high scale and multimodal, kind of looking at more than one type of DNA or RNA at a time. So that's kind of the first part of the presentation. The second one, I wanted to go over a few of kind of what I call like the next gen applications. So again, we've kind of in the last presentation, Doron kind of covered some of the applications that have been kind of being performed over the last few years, and really gaining a lot of scale even in the clinic. But some of these are actually taking a turn. Again, based on mostly sequencing costs going down, new methods of preparing samples, and then also a lot of the compute that's allowing us to look at data in new ways. So I'll start with kind of basic genetic genomic sequencing, which are really moving from panels to whole exome to whole genome. So now is probably the time where most applications are consolidating in germline sequencing around the whole genome. It's cheap enough to do at $100 that it's actually might be even cheaper than doing kind of a capture and then sequencing parts of the genome. And we're getting to the point where we can actually do that also very fast. And last year, kind of this presentation in ESAG together with the Broad, Roche kind of showed they can sequence a clinical genome for a neonatal disease in 6 hours. From library prep all the way to a VCF and a report for a physician, which is really what the field is looking for in this neonatal kind of rare disease scenario. The other side of the story is that it's actually not just about the sequencing. It's a lot of the problem is actually about the interpretation. So you can sequence the whole genome and you can find all the variants. There's thousands of variants in each one of our genomes. They're mostly private. Some of them are population-wide. But telling which ones are actually disease-causing is a very hard and unsolved or not fully solved problem. Probably something like 30–40% of newborns with a genetic disease, we can find the cause for that disease. In other cases, it's not clear. Either it's a kind of multi-genetic situation, or it could be that it's not in a gene. It's outside of the gene. It's in a regulatory region, intronic region, and so on. We don't really understand the genome well enough to understand mutations in the genic regions. And some of the work that's being done today, again, with the kind of the power of sequencing and high efficient sequencing, is looking in other dimensions to interpret and resolve kind of functionality of events. Two projects which I was involved in a collaboration with Ultima had to do with this kind of approaches for interpretation. One of them was looking into RNA, and the idea there was that when you find mutations, you can also look at RNA and see which expressed transcripts are carrying these mutations and whether it's actually affecting isoforms or expression levels. And the main challenge there is that you don't necessarily have the relevant tissue. If you're looking at neurological disease, the genome is going to be the same genome in neurons and in blood cells, but the transcriptome is very different, and you can't get a piece of an infant's brain to kind of see what the expression levels are like. And so what you can do is you can actually take cells that are easy to get, like fibroblasts, and sequence them to a very, very deep level, and you'll start seeing kind of abnormal splicing and isoforms, which are not kind of generally expressed in fibroblasts, but they can kind of leak even in these cells, and you can get like hints for what aberrant kind of expression looks like. Another type of orthogonal testing is looking at genome contact mapping through Hi-C, which is a method where you're actually looking at the three-dimensional structure of the genome. We tend to think of the genome DNA as a kind of a string of letters, but it actually bends, and there's like those knots and there's circles and there's loops, and a lot of the regulation happens in these kind of structures. When you do Hi-C mapping, you get information about these kind of structures in the genome, and you can see where they change. So using Hi-C to actually see kind of disruptions of regulatory aspects of the genome is another way to get kind of orthogonal data to resolve functional changes. Moving on into oncology, that's probably the area where most of the clinical sequencing is being done today. CGP is a kind of an acronym that was coined in the last decade or so for comprehensive genomic profiling, meaning that in one test you can actually take tumor tissue and test it for a lot of different types of events, mutations, copy number changes, translocations, and so on. It's actually becoming the standard of care for therapy selection, at least in late-stage cancer. So in many cancers, there's kind of a well-defined protocol for early stages, but once for the first kind of round of therapy, but once patients have exhausted these kind of options, when physicians are looking for like the next set of therapies to kind of use and try to kind of extend the life of the patients, and then using these kind of personal genomic profiling assays becomes very useful. And as we're seeing here, between 2000 and 2020, there's really been an explosion in data on the types of events driving cancer. For example, in lung cancer, we used to think of small cell, non-small cell, maybe a few drivers. Now we know of tens of different drivers. Some of them have therapies, others have therapies in development, but they're all kind of vulnerabilities of the tumor which you can exploit in therapies. And we've also, through CGP, we've actually also identified rare treatable events, and there's been kind of a lot of cases for like a personal case where a cancer which was untreatable turned into one that can be treated with maybe a therapy that wasn't even on the list. As for germline sequencing, in cancer as well, using whole genome and whole exome is becoming more of the trend. It's not happening as fast for two reasons. One of them is that for cancer you have to sequence deeper, so deeper whole genome is still probably not completely affordable, and also because most of what we understand in cancer still happens in genes and around them, and it's going to be hard to treat mutations outside of the regions we know about. But that's also happening. There's been some papers showing you can get better and more informative information by looking outside of the exome. And then kind of moving on to what's probably like the hotspot of sequencing oncology today, that's plasma sequencing, or what's called liquid biopsies. And that's actually using DNA from blood as a surrogate for sequencing cancer tissue, DNA from cancer tissue. And the idea is that we actually have a lot of DNA floating in our blood, cell-free DNA, which is coming from cells that have died, or otherwise there's necrosis, apoptosis, and so on. Most of them are coming from white blood cells, but some fraction comes from other cells that have shed DNA into the blood. In NIPT, we use that for newborn screening or prenatal screening, but in cancer, you actually get some between a tenth of a percent and tens of percents of the DNA in the blood coming from the tumor. And we can use this for different stages of disease monitoring to kind of find out what's going on in the tumor. You can use it for treatment selection, like from the tissue. But today, the main interest is in early detection and in monitoring response to disease, treatment. That's kind of where a lot of the current applications and commercial sequencing is being done. I wanted to kind of mention one of the technical aspects of this application. This is called MRD, so it's kind of monitoring the rate of residual disease in blood after therapy. So for example, you have a person with colorectal cancer, stage 1 or 2, they go through a resection, and the patient and physician want to know whether the full tumor has been resected and there's like no microscopical kind of tumor left, versus there is some kind of residual cells, and the cancer is going to emerge back in a few years. So one way of doing this, which is exemplified here, is taking the tumor, which you've just excised, genotyping it, and creating a patient-specific mutation list. So you know that this specific tumor has these specific mutations, and now you can use that in the blood to kind of look at these specific locations in the genome and profile them and kind of count whether you're seeing the same mutations that you saw in the tumor. And this is actually a very useful and proven way to track tumor progression or recession. And this has been actually developed. There's now clinical applications using this, like Natera and so on. There's a few companies developing these kind of applications, and there's been kind of an interesting version of this approach developed by C2I Genomics, which was later acquired by Veracyte, which is the whole genome version of this, which means you sequence the whole genome, but then you don't actually select the specific regions of interest. You actually sequence the whole genome in blood too. And from a statistical point of view, you can find enough mutations to kind of get to very good sensitivity. So essentially, you're replacing depth with breadth. And the nice thing about it is that you don't need a patient-specific assay. It's actually just kind of doing whole genome in blood for all patients in the same way. So it's a very, very generic and easy to run assay. Giving a bit of flavor of other types of assays that are being run and becoming clinical, we kind of mostly focus on DNA, but as I mentioned, there's other aspects of genomics which are interesting. So going from genome to methylome, which allows you to identify in each gene not just what the mutations are, but what are the cells of origin that this DNA is coming from. Since every cell type has a different methylation signature. Epigenome is kind of flags on the DNA that on the histones, and essentially tells cells what to express. So you can kind of see what the cell state is, and so on. You can look at transcriptome, which is expression, proteome, and so on. I wanted to kind of give examples of a methylome and epigenome analysis, which they actually allow us to take a blood sample and then look at it and say where the cells are coming from and what is their kind of expression signature based on the methylome and epigenome. Several different companies are kind of working on these kind of assays, which allow us essentially to look at different modes of disease and identify types of cells. For example, you can tell pancreatic cells and they're kind of how much they're kind of shedding into the blood. Patients with type 2 diabetes and type 1 diabetes actually look very different in that aspect, and so on. And that's all for methylation analysis. If you're looking at the epigenomic analysis, again, cells shedding DNA into the blood, you can see different types of cells, but also different states of the cells. So, for example, liver always sheds DNA in the blood. There's always processes happening in the liver, but you can actually tell apart patients which are having a liver-related disease for ones with healthy liver by looking at epigenetic states of DNA in the blood. And lastly, I wanted to mention one area that's kind of emerging. I think it's like super interesting. And again, it's really a consequence of the cost of the dropping cost and kind of high throughput and sample prep methodologies that are being developed. And this is using single cell analysis from blood to replace bone marrow biopsies. So two different studies. One of them is from the Weizmann Institute, and the other one from Dana-Farber showed that you can actually use cells from the blood to track different types of disease states, which today are actually being done from bone marrow biopsy. So on the left, it's looking at MDS, which is in kind of an early stage of pre-leukemia, and you can actually see in older people how cells kind of differentiate, and patients who are developing leukemia have actually very different profiles from ones that don't. And on the right, you can see patients with myeloma, and today they undergo bone marrow biopsy as part of their treatment plan, also for diagnosis, but also over time for monitoring. And in this study, the researchers showed that this can actually be done from blood by identifying cells that have CD138 markers, and you can actually get the same information, or even better, directly from blood. So that's a bit of just a taste of what's going on in the clinical world with new applications based on kind of new sequencing modalities. And maybe just to conclude all of this is kind of the idea also that the new kind of molecular diagnostic assays also provide optimal sources for data generation. So if you can imagine, like in this last slide, we're looking at thousands of cells from thousands of patients. It's a very, very rich data set, and for each one we have like full transcriptome. It's a very rich data set to be able to kind of look at new disease modalities and applications. Thank you.
Thank you very much to both of you. Thanks very, very much. I'll open up for questions. Please raise your hands. Either use the QA box or raise your hand. I'm not sure if I see if people raise their hands. Do I see that?
There have been some hands. I don't know who.
Sorry?
There have been some hands floating around.
Okay, yes. As I see no questions from the attendees, I have some things that really interest me, and you really seem to be at the source of that. And because what many of us are working on is DNA data storage, and there we have cost is a major bottleneck. As of many things you've shown, Doron, you've showed about these things and how, let's say, changing the cost limit enables new applications. We have a second cost limit, which is synthesis, but if we just focus on sequencing, how do you see this moving in the future? It seems there are more and more, let's say, if we lower cost also from the biological side, there will be more and more applications that can use sequencing. So what's let's say direction that can go? And this cost, is that a real technological cost, or is that a cost market fit kind of, let's say, let's assume that I could offer whole genome sequencing for $1. My company couldn't make enough money because there's not a market large enough. So where is the, let's say, technical limit, and how do you see that, let's say, that the current cost structure is going to develop in the future if we assume there's no DNA data storage? Let's say if DNA data storage would be a thing, we could really flood the sequencing market with massive applications. Yeah, so really if we think about going into the cost direction.
Yeah, I think there's a few aspects of that question that are worth considering. One of them is that, as you mentioned, sequencing costs can continue to go down, but at some point, and we're actually pretty close to that point, it's not the major cost driver for a specific application. If you think about single cell, whole genome, single cell, whole genome methylome, and so on, whole genome cancer genomes, for example, you can kind of see that sequencing is actually probably at this one like a third or even less of the sequencing cost. If you think of a clinical application, if you add on top of that everything that has to happen to actually for a patient to get a report, it's actually even less than that. So driving sequencing cost even lower starts to become kind of irrelevant. And as you're saying, it also kind of makes, it's also, the main thing it does is make the sequencing part, piece of the pie kind of lower. So there's even less incentive to drive the cost down. I think it's kind of a chicken and egg thing, because as these costs go down, there's more applications that use more data. And you can imagine that even for a single sample, you could get more data. So very kind of data-rich applications kind of become more possible when sequencing cost goes down. So I think there is room to continue to push on cost. It'll happen as part of a kind of a whole ecosystem kind of drive for end-to-end application to become lower. So I think kind of that's kind of the main part of the discussion. You know, there is a belief that, you know, there's enough elasticity in this field that when assays become cheaper, then people do more sequencing. In the research market, I think that's very true. People have grants and, you know, they have budgets and they'll use them for doing these assays. And if the assay is cheaper, they'll probably look at more samples and more data. In other areas, it's not exactly the same way, but again, I believe that new applications will kind of emerge that use more data. But again, that takes time. I think it's probably going to be like a longer cycle than what you see in like consumer electronics.
Yeah. And then, Laurent, if we go back to, let's say, 15 years ago or 20, if you think about what were the discussions of cost at the time, how far away was, let's say, $100 genome from what you envisioned at the time, or how were the discussions at the time, how cost would develop in these technologies?
Well, I think Doron has shown really much with this beautiful graph on the cost, and we envisaged that it would go on the speed, and now we see that there is a kind of threshold that we cannot go too easily above that. And I share the view of Doron that today the major driver of the cost is really around. You have two things in medical diagnostic: is all this regulation with CE-IVDR, which is making a lot of work to ensure that we have protocol. It's slow, the process where you can release new technology because it takes so long so that you can apply it in a diagnostic field. And if you have the commercial application, you cannot do in-house. And that's a very, very important driver. And the other one is, of course, the interpretation. Now with this tremendous amount of data, we need to have like artificial intelligence to mine the data, to find the relevant variants. We have clinicians who are working together with the lab people, and they are spending a lot of time to find what is relevant. And also another problem in the clinical field is what to show, what to give to the patient. Because imagine you have a couple waiting for a baby and you tell them, look, you're looking a little bit of, missing a little bit of chromosome 17. We don't know what it is doing for your baby. What do you decide? Ethical part will be also an important part of this discussion for the counseling. So that's probably the cost is not on the sequencing itself. And as I mentioned, probably sequencing or resequencing is cheaper than storing on the computer or memory. And that's probably your project. But all the rest in clinical application is taking more. When we have the cost structure now, sequencing is only a small part.
Okay, but then if we think as a sequencing company, DNA data storage would be an interesting avenue, because we neither have the problem of, let's say, data handling, or we know how to, let's say, that we have pipelines to handle that. And also the ethical question seems to be much easier if we go to data storage applications. So in terms of a sequencing company or technology who wants to continue to push sequence technology, that would be an interesting aspect to evade, let's say, all of the complications that come from the medical field.
Yeah. Another part is you cannot hack the DNA stored.
Yes.
Hopefully.
Yeah. I think there are several applications. Yeah. But really, in terms of sequencing, I think that there's really, that would be of interest. Second thing I found really interesting, both of you never really talked about Oxford nanopore technologies, where I see in the scientific world, we see a lot of interest in that. It's so small and so let's say, flexible. And Doron, you kind of excluded that from, let's say, it wasn't in your examples. So how do you see the space, or let's say, of Oxford Nanopore in, let's say, the really medical applications of sequencing technology?
And I'd like to add to this question, actually, to both of you, in terms of modifications in RNA or in DNA. So Oxford Nanopore is the company that claims to be able to do some of these. And how important do you find these to be? Obviously, Laurent, PCR-based or amplification-based approaches cannot do it. So what is your perspective on that?
Yeah, so I mentioned in the beginning as a third generation technology. I think the reason I didn't focus on it so much, again, I could have added another modality, is I don't think they've really come out of like a very niche space yet. I know there are companies that are using it for kind of specific applications, and you can say, this kind of very small, like USB type kind of sequencer, has application that only it can do. That's the only one you can take in a jungle and sequence kind of your specimen on the spot. So I think it is interesting. There are some clinical companies looking at that, and again, I do come across it once in a while. It's usually kind of a way to fill in gaps, things like that. I think there are other. I think PacBio also measures kind of 5 bases. So again, the single molecule sequencers have that advantage, and so I would kind of couple that into that bucket. It'll be interesting to kind of see whether the Roche sequencer can do that as well. So again, I'm sorry, I even gave the asterisk that I'm not covering all sequencers just because of time. If in a longer kind of review, I would definitely include.
I just see the mismatch that a lot of, let's say, we find this real nanopore, let's say, I think it's scientifically interesting because it has some problems in, let's say, base calling, which are really interesting in solving. And really the compactness makes it so appealing and, let's say, low entry cost for scientific applications. But it seems, if I listen to you, if we go to, let's say, medical applications, the advantages it brings don't outweigh the cost it has, or let's say the disadvantages it
I would
brings.
just say it seems to have been that way so far. I wouldn't say they can solve it, but they've been around for a long time and haven't like really broken into the clinical market other than in kind of like the fringes. I definitely see people using them for like filling in gaps. It's usually like the second sequencer. It's not the main one. But I'm excited to use nanopores as much as any other sequencing enthusiast.
Then I have 2 questions from our audience. One from
maybe
Laurent, can you add your perspective on modification?
Yeah, we are working with Oxford Nanopore and using it in the clinical field. Our colleagues are thinking it's interesting as a complement. So as an additional sequencer, and same with the PacBio. For example, the new Vega released a carrier screening panel that is very interesting for difficult genes that would, in the clinical space, need Sanger or MLPA to analyze these difficult regions. And with the long reads, you can address that. So there is strong interest of our colleagues for this application, and we are working on that.
And how about modifications, methylations, or RNA methylation or acetylation that are ubiquitous in the genome? We know that, but do people try to use them at all in the clinical space at this point?
Yeah, they do. We have been using that for a long time, and now with this new application, it's much easier. And we are also working with colleagues on epigenetics and these technologies. Of course, it could be based on arrays, but although sequencing, it's a lot of interest now. There is for epigenetics, a lot of interest also for the well-being, not only pure clinical genetics, but simply the well-being of the people.
So which technology are you using for that with customers in the company at this point?
For the moment, it's a lot of microarrays because it's easy for these applications, but we are using also PacBio, for example, for this technology.
Thanks.
Then I have 2 questions from the audience. One is from Ashern Istfanfi. Hi, thank you very much for these great talks. I really enjoyed them. One question, is there an interest for Illumina sequence to incorporate the detection of non-canonical nucleotides as well? So do you have any insight in, let's say,
I don't know.
non-canonical nucleotides?
Don't know.
Doron, also any of those technologies, do you know
No,
anything
I
about?
think I think two things to think about. I don't think that a lot of them are doing it right now, but if you think that if this would to become a significant kind of need, definitely like single molecule sequencers like PacBio and on Oxford and Roche could enable that. So again, I think it's not that it's happening by itself, but were it to become a need, you can imagine there's a way to do it.
Okay, thanks. And Jörn Glöckler asked, will there be a way to sequence, let's say, 1,000,000 batches, each comprising 1,000 or more different molecules in parallel without barcoding? So a highly parallelized batch sequencing kind of. Do you see that in, let's say, coming?
So why not use barcoding?
Sorry?
Why not use barcoding? I'm trying to kind of understand the question. You have to separate the batches somehow.
You have
You
to
could do it,
separate
you could do it
barcoding, let's say,
temporarily,
yeah, adds
or you could.
complexity, let's say on the, let's say, DNA side, it would shift that complexity to the, let's say, sequencing side. Also, if you think about, let's say, our DNA data storage world, time also becomes an issue. So often I see that less of a problem in medical applications, but often everything we do on the DNA is relatively slow compared of things we can do on the sequencer, which is relatively fast. So do you?
One thing I would think about in this context is single cell, and people have been doing kind of million cell experiments and even more than that today. And I would say it's not without barcoding. That's kind of why that constraint may create some challenges. But you can barcode 1,000,000 cells or 10 million cells and so on in a way that you can read all of them and all the transcriptomes. So it's probably more about how do you kind of come up with a clever barcoding or some labeling technique so that you can read it. Think, the sequencer can read billions, tens of billions of pieces of DNA. The sample prep is what determines what it reads. So it's not so much a sequencer problem than a prep problem. And maybe
the
think about how
data
to actually use cell machinery to do that.
and the data analysis question, how do you tell them apart? I think that's part of the issue. I mean, you can, of course, mix them, but then how do you tell them apart? That's part of the, I think, of the analysis challenge maybe here.
Yes, also I know Laurent did quite some work on barcode spillover, is that correct? Or let's say misreading or let's say misintegration of barcodes. Laurent, what's your view in, let's say, barcoding and trying to avoid it, or let's say alternatives to barcoding, or let's say solutions to some of those problems in barcoding?
Yeah, we developed a lot of barcoding for the metagenomics application with different levels of barcodes, but well, it works pretty well. So I don't see too many challenges in that.
Yeah. So you would both say that barcoding is the best way to go
Yeah, and definitely, methods for error correction. I'm sure if you're thinking about these things, a barcode doesn't have to be like a random set of letters.
Yeah.
No, so I think you're now added to the question. I think in the context of storage, that's true. If we can not use barcodes, it will help us. And there you could separate them just by the payload sequence. I think that makes sense. And for the context of data storage, I think this will be useful. Yeah, can be useful.
Yeah. Let's see if I see more questions. Yes, then I have from Fredrik Walter. Thanks you for the interesting insights. What are the main users? What are currently the main users of sequencing technology? Is it still more research or standardized use tests for medical tests? So how in volume is, let's say, medical applications over research applications? I think, Doron, you hinted on that.
Yeah,
Let's
I
say
don't know the
the
exact
funding
numbers.
background.
Yeah.
So yeah.
I don't know the exact numbers. I think today, if you look just like at bottom line of sequencing vendors, I think clinical is more than research, probably, but probably not by like a factor, probably like 2 or 3 times higher. And it's driven by like a few a few very large labs that run a lot of sequencing. And again, it's cancer, genomics, and so on. But there's a very, very long tail, right? So when you're going to research, lots of like smaller studies, smaller labs that are doing a lot of work.
Yeah. And Laurent, in let's say, what do you see in Switzerland? You're more in the medical space, I assume.
Well, and no, we still continue to do a lot of service for researchers, and of course, you can have small lab universities, but we have a big volume from pharma company or agro biotech companies who are doing like
In
field
research,
biopsy,
yes.
thousands of plant samples, and
Yeah.
this is high
Yeah.
volume.
Okay. So yeah. It's more, so if I understand correctly, Doron, more, let's say, medical, but it's still the research still has a large fraction.
Yeah, and you know, it also depends where you put translational research, right?
Yes.
Like Laurent said, pharma does a lot of sequencing, and it's, you know, they'll think of medical applications, but it's not necessarily a clinical application yet.
Okay, super. I have no more questions. I see none of our attendees. Toha, anything you want
I
to?
have one more question. So the one you mentioned that it's sort of becoming cheaper to do whole genome than to do panels. So I guess the question is for both of you. First of all, how do you reconcile that with the need to have a lower limit of detection? If I want to find a variant which is 1 in 10,000 or something like that, and there are applications like that, I will still need to focus, I suppose. And the question to Laurent is are you seeing that as well? Are you thinking that from the customer's point of view, they will want to do whole genome rather than panels in monitoring specifically?
Yeah, so obviously it depends on the application. Germline sequencing is kind of where it's moving the fastest because 30x whole genome is becoming cheaper than any kind of selection. For cancer genomes, it's not true yet because doing a 500x genome isn't cheaper than doing a whole exome. And I think monitoring is actually the interesting area because you're not actually looking for a specific variant. And this is the example I gave with MRD. If you're looking for a signature, it might still be cost beneficent to do it in a whole genome way or not, depending on a lot of parameters of the problem.
But you're looking for minute frequencies still, right?
But you can, again, there's papers showing you can detect 1 in a million by looking at the whole genome with enough mutations, right? I mean, you can probably do the math to figure out. It's not true for every tumor, but it's true for most tumors.
Yeah. And on our side, it's pretty much the same. So we kind of abandoned the small panels. We have a single pipeline, which is accredited. It takes a lot of time for all the accreditation work. And we do a whole exome, and then we have in silico panel for other application, germlines, of course, but also now we're switching for the somatic panel because we have enough depth for the tumor analysis. And we also have requests for whole genomes, especially using the PacBio technology. So we, as a clinician, they start to ask for whole genomes. It's difficult because of reimbursement. That's the challenge we have with the insurances. The price of exome is still relatively high. We know it will be reduced at one time. And of course, that's another of the challenge. We need to have each time the okay from the health insurance that they will pay for the case, and the doctor have to ask that for each of the patient.
Thanks. That's not good news for synthesis companies, by the way, but it's good news for science. Yeah.
Yes, with that, we're already well over time. I think this is a really interesting topic, this intersection of, let's say, real technology and medical field, and also the cost effect that goes into that. I would like to thank Laurent and Doron very much for your talks and the inputs you gave us during the discussion section. Thanks very much. Hope everybody has a very nice day and see you all again soon. Bye-bye.